This is the lecture for Chapter 10. Some textbooks introduce recombinant DNA technology piecemeal throughout the book as they talk about different concepts in cell biology. This book has a whole chapter dedicated to it, so this lecture is just going to be about some laboratory techniques that are useful in doing research and that impact everyday life somewhat, and the technological advances that they bring. So the earliest advances in DNA technology came from isolating distinct fragments of DNA and cloning them. Cloning just means to make identical copies of something. So cloning DNA is to make multiple copies of that identical piece of DNA. Having a distinct fragment of DNA that you're interested in is great, but it's only useful if you have it in great enough quantity to work with in the lab. So from the very beginning it became necessary to not only isolate DNA from living things, but also to make many copies of little pieces of their genomes that are interesting. And the way that this became possible is through a seemingly arcane finding about bacteria, which is that if you present bacteria with DNA in the lab, they will cleave it and they will chew it up, and they will do this at specific nucleotide sequences. So because these enzymes restricted the transfer of DNA between bacterial strains, they were called restriction endonucleases, or what's more commonly just called restriction enzymes. An endonuclease, just so you're clear, is an enzyme that will cleave within a DNA sequence. An exonuclease cleaves only at the ends. So restriction endonucleases cleave within DNA sequences. And as I said, they cleave at specific sequences. And this is just 3 examples of this. So HEY3 will cleave at this GGCC sequence, and it's a blunt end cutter, so it cleaves right in the middle, and these are the products of how it cleaves. EcoR1 cleaves this particular sequence. It leaves sticky ends. The TTAA, AATT is what's left over, and HinD3 is also an enzyme that leaves sticky ends. One thing that's interesting about restriction enzymes that you might notice is that they cleave at palindromic sequences, which is sequences that are the same read from 5 to 3' and the same read from 5 to 3' on the other strand. So GAATTC is the same as GAATTC. These are palindromic sequences, and it's a feature of restriction enzymes. Another thing to notice about these is that they are named for the bacteria they come from. So EcoR1 comes from E. coli and so on. There are hundreds of these that have been isolated. They can be purchased from a catalog, from a biotech company, and they're very useful. In fact, they're sort of critical for DNA cloning as we know it. So the second important component for recombinant DNA technology are the vectors. A vector is another name for a plasmid. They exist naturally in bacteria, and they're sort of a mobile supplement to the chromosome. As you know, bacteria have circular chromosomes, and that's where the majority of their genome is located. That's where all the genes for doing metabolism and growth are located. But they also have, and by the way, the chromosome is a single copy, but also within bacterial cells there are many copies of small pieces of circular DNA called plasmids. Often these will have resistance genes for antibiotics and things like that, and they can be transferred between bacteria through a pilus, also known as horizontal gene transfer. Plasmids are useful because they can be isolated from bacteria. They're in high copy number, and they're easy to handle, and they can also, of course, be cleaved with restriction enzymes. So in this example, it's showing vector of some kind that's cleaved with a restriction enzyme, and here's a electron micrograph of that. A DNA fragment has been also cleaved with the restriction enzyme, and assuming that they have the same sticky ends, they'll be joined together, and then you add DNA ligase, which is the very same DNA ligase that you've heard of before in DNA replication, and the two pieces of DNA will be joined into another circular DNA molecule, and it's now called recombinant DNA. It's called recombinant because it's combining DNA from two different sources, from two places that they didn't ever exist together before, and now they do because they've been brought together in the lab using these techniques. So what happens next is that the recombinant DNA needs to be introduced into a bacterial cell through a process called transformation. The bacteria need to be competent to take up DNA, which means they've been treated with calcium chloride, and then the recombinant DNA is added, and the bacteria are shocked into taking it up, either through heat treatment or through an electrical shock. Anyway, once they've taken up the DNA, then they're put into culture media that allows them to grow and produce millions of copies. And from this culture, plasmid DNA can be isolated, and in great quantities. So sometimes researchers want to work with a single gene or a stretch of DNA of interest, but oftentimes they're interested in more than that, and sometimes they're interested in the whole genome. And when that is the case, the whole genome will be digested with restriction enzymes, usually more than one, generating millions of fragments, and these fragments are inserted into plasmids using DNA ligase, as you've seen, and then put into bacteria. The bacteria will be in a liquid culture, and they'll be, selected for the ones that are possessing the plasmid, because not all of them end up taking a plasmid, but the ones that have a plasmid are selected for using an antibiotic resistance. And then you have sort of a flask of bacteria that has the entire genome in it, in plasmids contained by individual bacteria. So I've always thought the word library is a little confusing, because to me a library is a sort of a repository for books from many, many different sources that come from different authors and different publishers and different places. But a genomic library all comes from the same place, right? It comes from the human genome, or the dog genome, or the yeast genome. So I've always thought that was a little bit of a troubled analogy, I guess. But I guess it works because, all the clones in the genomic library are like millions of different books, all with unique content. So in that sense, it makes some sense. But in any case, it's called a genomic library, and this is how it's made. This is what it's composed of. So, libraries are useful for lots of things. One thing they're useful for is to sequence a genome. You can pull out individual clones and sequence them, and then use computer algorithms to compile your sequence into contigs, and then eventually sequence the whole genome. They're also useful for identifying genes of interest from the protein. Sometimes an interesting protein is purified in the lab using biochemical techniques, running it on columns and testing it for activity, and then eventually having a pure protein that's interesting. And that pure protein can be sequenced, so its amino acid sequence is known, and then the likely DNA sequence that it's coming from can be predicted, and that can be used to pull out the clone from the library that has your gene of interest. So how is that done? It's done through this process called hybridization. Hybridization means I'm taking a piece of single-stranded DNA and I'm matching it to another piece of single-stranded DNA that is complementary to it. So hybridization means I'm matching up complementary single-stranded DNA. Okay? So the way that this would be done is to start out with a genomic library and to plate it out on petri dishes. This figure is showing one petri dish, but in fact you would be doing this for several petri dishes, and they would have hundreds of thousands of, not hundreds of thousands, but thousands of colonies on them. Anyway, each colony comes from a single bacterium with a single recombinant plasmid in it, containing a single stretch of DNA. Those colonies are blotted onto a piece of paper, and then the paper is treated in a way that opens the bacteria so that the DNA is still there, and then the DNA is fixed onto the paper so it won't sort of slide off or get washed off. And then you take a probe for a stretch of DNA that is of interest. So you could say, I'm interested in gene X. I have a little piece of its sequence, and I'm going to tag it with radioactive phosphate, and this is my probe. And then I will wash my piece of filter paper with my radioactive probe, and then wash it off. And then any clones that are containing my gene of interest, the radioactive probe will stick to it because the two sequences will hybridize. And then using photographic film that will be, developed by the radioactivity, you can see which colonies in fact have the gene of interest that you want to find. And then you can go back to the original dish and pick that colony and grow up many copies of it and isolate that DNA in great quantity and then work with it. So sometimes a genomic library is really useful, but sometimes another kind of library called a cDNA library is useful. cDNA means complementary DNA, and it's made from mRNA in the following way. mRNA is isolated from cells, and you provide it with a primer that's just a poly-T that will hybridize to the poly A tail of mRNA. And then an enzyme called reverse transcriptase will make a DNA copy using the RNA template. And the RNA is removed and you're left with single-stranded DNA, and then DNA polymerase can use that as a template to make double-stranded DNA called cDNA. The cDNA can be inserted into vectors, just like genomic DNA can be, to create a cDNA library. And cDNA libraries can be revealing in the following ways. Depending on what tissue you started with, some genes will be present in your cDNA library and others will not, because obviously not all genes are expressed in all tissues. So that it can inform you about the tissues that are expressed. It can inform you about expression, about time and place of where different genes are expressed. And it can also be informative about where introns are located, because if you compare the cDNA to the genomic DNA, it will tell you where the introns are.